All right, here we go once again. Let me remind you quickly that last time what we did when we talked about DNA replication was we talked about what if you had a cell like this one here, for example. This is the nucleus of a cell, but let's say you had a cell. You wanted to copy everything in the cell, so you could divide that cell, and then you would have two cells that were identical to that. That was called DNA replication. What we're going to do today is we're going to talk about what if you have a cell and that cell is trying to make certain enzymes. Let's say you drank some milk, and inside the milk, milk has a sugar called lactose in it. And so let's say you wanted to break that lactose down. Your body has in it DNA that has the instructions on how to build an enzyme called lactase. For example, your body can make that lactase and it could break that sugar down and digest it. And that's true of all sorts of things that go on in your body. So what we're talking about today is how we can take that DNA and how we can use pieces of that DNA from one section to another to copy just a piece of that DNA as the instructions on how to build a protein. And so this is different than DNA replication. So we're just going to copy one piece of the DNA here. When we copy one piece of the DNA that's used to make a protein, we call that a gene. So you've heard of genetics before, or a gene. You know about genes. Your genes are essentially the instructions on how to build proteins. Okay, and so in a human, we used to think maybe originally about maybe 20 years ago that there were probably something like maybe 100,000 genes in a human, and now we think that order is somewhere on the order of more like 30,000 genes in a human, somewhere in there. So what we're going to do is we're going to talk about two different parts as we talk about this, and that's called transcription and translation. The first part we'll do is called transcription, and in the case of a eukaryotic cell, that's going to take place inside the nucleus. So in a cell like yours, this process, transcription, takes place in the nucleus. So this is the nucleus here, this is the cytoplasm down here, and this whole big thing would be one cell. So we're going to take that cell, go through transcription, and we're going to make RNA, and then the RNA is going to come down and be read by ribosomes in a part to make a protein. If you remember ribosomes way back, maybe on week 2, we talked about the nucleus being the brain of the cell, probably is how I like to refer to it, and then protein or ribosomes are used for making proteins. And now we're going to put all that together and see how that's done. Okay, so first of all, let's talk a little bit about DNA versus RNA. So we have DNA and we have RNA, and for the most part, they're quite similar. They have a nitrogenous base, and they have a sugar, and they have a phosphate group. In RNA, remember that RNA, we have the sugar ribose, and in DNA, we have the sugar deoxyribose. Okay, and there's the difference right there. You notice this has this one hydroxyl group there, the OH, and this one has a hydrogen there. So that's one difference. The other difference is in DNA, most of the time DNA, there's exceptions with viruses and things like that. We'll ignore those now, but just in case you ever come across, if you take micro, for example, you might see this. But DNA tends to be double-stranded, it has two sides to it. RNA, again, not necessarily true in viruses, tends to be a single strand like that. So that's one difference as well. And then also, you notice that RNA has, in addition to having cytosine and guanine and adenine like you find over here, instead it has this nitrogenous base called uracil. And uracil is found in RNA. Thymine is found in DNA. So the uracil will now bind with the adenine instead of thymine if you have RNA versus DNA. So that's the difference there. So what we're going to do is, once again, since this is Bio 1, we're going to skip some of the proteins that are involved in initiating this step, and what we're going to do is we're going to just start at one point and say, okay, what happens is we have this enzyme called RNA polymerase. which is kind of similar to DNA polymerase that we mentioned before. So here's my RNA polymerase, and what RNA polymerase is going to do, it's going to start at one section, and it's going to copy one side of the DNA. So it's going to bind to it, and it's going to start reading that DNA, and as it moves along, it's going to hook nucleotides together. So here's my DNA, all right, guanine, cytosine, adenine, adenine, so forth, and this is my, what's going to be called eventually my mRNA, which is my messenger RNA. Okay, but first we're just copying my DNA into RNA. So you notice there's my G, so that's going to bind with a cytosine, and then my C is going to bind with a G. And notice once again, as I mentioned before, if I have adenine, instead of having thymine, since this is RNA, I'm going to have a uracil. So uracil there, cytosine. Notice I have, here's my DNA, so there's thymine, that's where adenine will bind. And you'll notice some are long and short. That's because remember that the guanine is a purine, and the cytosine is a pyrimidine. We mentioned that before last time. Okay, so what's going to happen first in the nucleus is I'm going to copy my DNA into the form of RNA, messenger RNA in this case. Now, what happens in cells like ours, you copy the RNA, but it turns out you don't use the whole RNA. So we have things that we call exons in there, and we have things that are called introns. So what will happen is, this is your original mRNA strand that you just copied. Some of these pieces get cut out. We used to call this junk DNA and junk RNA, like we didn't need it, but it turns out maybe this actually does do stuff. But what we're going to do is we're going to take that exon and that exon and that exon, and we're going to hook those together. That's going to be my original code. So there are enzymes that will fold this up, and they will cut out the introns. And a colleague of mine, I have to admit, came up with this, but if you think about this, the introns go in the trash. Introns go in the trash. So we get rid of the introns and we keep the exons. And so you hook the exons together, and once we do that, that's called my mRNA. So this process here is called RNA editing. So it doesn't happen in all cases, in all cells, but it does in cells like ours, for example. So the introns go in the trash, and you keep the exons, and that becomes our mRNA. So if we look at that, then what we're going to do next is we took our DNA and we copied it into RNA, and then we cut out the introns. Those went into the trash, and so now this is my edited piece of DNA. And in a living system, there are 20 amino acids that are used to make proteins, 20 amino acids. And you notice that we have only 4 nucleotides. We have guanine, we have cytosine, we have adenine, and we have thymine, or in the case of RNA, we have uracil. So we have 4. So what we need to do, in order to code for all 20 different possible amino acids, we need to read the mRNA in sections of 3. So that's what we're going to do here. We're going to read them in sections of 3. So I'm going to read my CGU, for example. That's going to code for the amino acid arginine. And we're going to go through that code, and that's the DNA, therefore, is going to tell me how to make my mRNA. My mRNA, then, when I read it, is going to be the instructions for the order in which the amino acids need to go together. That process is called transcription. So, I'm sorry, transcription. I have transcription twice on there. Okay, don't know why that is. Let's cross that out. I read it correctly. Luckily, I have this in my head. So transcription, maybe they messed up this picture here somewhere along the line. But anyway, what we're going to do is I have my DNA, and I'm going to copy my DNA into mRNA. That's called transcription. But when I read my mRNA in sections of 3, that's going to be called translation. And one of the ways you can think about this is when you transcribe something, you're taking it like from, I used to work in medical records, for example, and doctors would have to transcribe the notes they took. So what they would do essentially is they would take a bunch of notes and then they would go call on a phone or have someone type out for them what they actually did. So they were taking their language, their medical language, and converting that into a typed form in that same language. So when we go from DNA to RNA, we're going from nucleic acid to nucleic acid. So we're transcribing it from one kind of nucleic acid also to a nucleic acid. But in translation, we're changing it. We're going from nucleic acid to amino acid. It's a different group. It's a different language. And so we call that translation, not transcription again. So it should be transcription in translation. The next thing we need to know is we need to talk about this other molecule called a tRNA. And the tRNA stands for transfer RNA. And transfer RNA is, once again, it's an RNA molecule that you see there folded up. On one side, it has a series of 3 RNAs that are going to be the opposite to what my codon sequence is, my section of 3 in my mRNA. Okay, so that's going to be called the anticodon, and that's going to bind to the codon, which is a section of 3 mRNAs. So that's on one side. On the other side, I'm going to have my particular amino acid. Okay, and if you look at this picture here, this shows you the genetic code based on what your mRNA would be. So this is an mRNA chart. So if you know what your mRNA is, for example, you can figure out what your amino acid would be coded for in that particular case. So if you notice here, we've got leucine and we've got leucine, for example. So if my DNA, okay, if my original sequence of DNA that I'm using was, say, GAA, all right, so there's my DNA. So what would happen is my mRNA would come along and I would code for that. And the equivalent to when I read that with my RNA polymerase, if my DNA is GAA, my mRNA is going to be CUU. Because remember, there's no thymine in RNA. So instead of having a T here, the base pair with the A would be a U. So I'd have my U binding with my U just like that, my G with my C. So my mRNA would be CUU. Then I would go, okay, what would that code for? What's my protein? Or what's my amino acid? Because the protein is all of those connected together. So what amino acid would that code for? Would be my question. And I look over on this chart, and what you do is you go, okay, here's my first base pair, here's my second, and here's my third. So my first one is a C, and my second one is a U. So that puts me in this ballpark here. And my third one is a U. And so that gives me then leucine. So leucine would be the amino acid coded for there. Now, what happens if you have a mutation? Mutation, again, is any change in the DNA. And you'll notice that I mentioned somewhere along the lines, I believe, that you could change the DNA, and sometimes that's good, sometimes that's bad, and sometimes that doesn't do anything. So, for example, imagine if I changed this, if there was a mutation that occurred that made that a C instead, then I have GAC. So I would have instead, my mRNA would be GU, and that wouldn't be a U anymore there, that would be a G. So I would have CUG. But you notice if I go down here, it turns out that even though that mutation would change the DNA and it would change the mRNA, notice it gives me the same amino acid, which is good because that means that I'm still putting the same amino acid in the right spot. So often we call this the wobble effect. So the idea of the wobble effect is that oftentimes that third amino acid, that third mRNA, what am I trying to say? The third nitrogenous base in that codon sequence. That third one, often if you change it, will still give you the right amino acid. And therefore, if you have the right amino acid, you still build the sequence correctly, and that's good because the protein probably will still function. Okay, so notice that, once again, let me point out, this is mRNA. Okay, this chart's based on mRNA, and it shows you which amino acid you would put where. Okay, and notice that there's several ways you can get leucine. For example, in this case, you see right here, we have 6 different ways we can get leucine. Over here, we have arginine. We have 4 ways there, and then we have 2 more ways there. So there's several different ways. There's redundancy in the code. There's several different ways you can get there. Notice we also have a start sequence. That tells you when to start building the protein. And we have stop codons as well, which tells the protein that's where you're going to stop building it. Okay, so let's kind of put all this together now, and I'll show you here. Here's my mRNA. So I build my mRNA, and that happens in the nucleus. So here's my cell. All right, there's my nucleus, and I build my mRNA in there. And then that mRNA comes out of the nucleus, and it's going to be read by ribosomes, and that's what you see here. So what's going to happen is, here's my mRNA, and I have 2 spots on this one. I'll show you a third one in a minute. But we have my P site and my A site. So what happens is, things come in, the tRNA comes into the P site, and the anticodon binds to the codon. So again, those are opposite. So that's in my P site, and then my A site comes in, I do the same thing. And what's going to happen then, once my P site and my A site have my tRNAs in them, and you notice this has been kind of coming along here, and it's already transcribed this part, and it's moving this direction here. What's going to happen next is this is going to float in here, and then all of this here is going to transfer over to this one. And then the whole thing's going to move that way, and I'm going to continue on. Okay, so if you look at this picture here, all right, here's my P site, here's my A site. There's a third site over here. They don't mention this in most Bio 1 textbooks, so I'll mention it because it comes up every now and then, called the E site or the exit site. Essentially, this whole thing's moving that direction. So what happens, what I was just telling you here, here is my P site, here's my A site, here's my codon down here, that's on the mRNA. Okay, that's my messenger RNA. And so that's my P site, that's my tRNA with its amino acid. This thing's opened up, so it's going to float in there and connect. And then there's an enzyme that's going to take that and move it to this one. So the amino acid from this one's going to move to this one. So I'll have 2 over here then. Okay, there'll be methionine over there like that. And so what happens, this then loses its amino acid, and this moves this direction and floats out. So now I have the P site open, I have the A site with my tRNA like that. What's going to happen then is this whole thing, including the mRNA, is going to move down that way, one direction. And that's going to open up my next codon. So the whole thing's going to shift down. And this is then, what's in the A site now is now going to be in the P site. What's in the P site is going to go to the E site, and that's going to float away. That's going to open up this sequence. This whole thing's going to move down one, and I'm ready for something to come into my next A site. Okay? This shows you that a little bit better there. So there's my P site, there's my A site. I've transferred the methionine over to the leucine. This is now open, so that's going to float away. All right. Now notice there's nothing in the P site. This thing went to the E site and floated off. And then this gets recycled. So an enzyme will come in, build another one of those, attach that, and you can reuse that one. So now my A site has my tRNA, it has my leucine, it has my methionine. Nothing's in the P site. This whole thing here in the A site, including the mRNA with it, this whole thing's going to move down one codon. Again, a codon is that section of 3. Okay? 3 nucleotides in a row is my codon. I'm going to read these codons with my anticodon. The whole thing's going to move down. That's going to open up this next one. So notice in this picture here, this whole thing moves down one. So that whole thing is moved down one like that. Now my esite's open again, and my next tRNA can come bind here. This then will transfer over to that, and we'll keep doing that until we're done. And that will build my protein. This is a picture sort of putting it all together, trying to show you the whole thing together. So here's a cell. Here's my DNA, here's my RNA polymerase molecule here, and again, I'm reading my DNA. Once again, just like in DNA polymerase, RNA polymerase travels in the 5' to 3' direction. So there's my 5', there's my 3'. RNA polymerase is like DNA polymerase, but it hooks RNA nucleotides together. So as I travel along, I'm hooking my RNA nucleotides together. That goes through RNA processing or editing, where I take the introns out and I get rid of them. And so what I'm left with is this mRNA. It has a whole bunch of adenines on one end and this molecule called a 5' cap on the other end. And so that's my mRNA. It comes out, and I'm reading now from 5' to 3'. I'm reading my mRNA molecule, and here's my tRNA. Here's that E site I told you about. This missing. This is a majors book, so they have the E in there. But here's my P site right there. There's my A site right there. And so as these tRNAs come down, what's in here, these are my amino acids. Those transfer over from P site to A site, and then this whole thing moves down one codon, and I read that in sections of 3. And as I mentioned, the recycling part, you don't need to know this, but here's amino acyl tRNA synthetase, which is basically building, it's an enzyme that will build new functional tRNA molecules, so it's attaching new amino acids to that as we go. And again, that whole process there, that involves transcription. And transcription is taking DNA and copying it into RNA, and also translation. And in translation, we take mRNA and we read the codons to build proteins or amino acids. And you hook the amino acids together, and the amino acids have to be in a particular sequence, because what happens is, if you look at this as a good example here, this amino acyl tRNA synthase, for example, it's an enzyme, and what happens is this folds up into this 3-dimensional shape, and you have to build this 3-dimensional shape exactly right for that whole thing to fit in there. So that this space becomes what they call the active site of the enzyme. If you remember, we talked about that before. And so if you have an error in the DNA which messes up the mRNA, which gives you a different sequence of mRNA and therefore different amino acids, this isn't going to fold up in the correct way, and it's not going to work properly. Okay? So again, this is transcription and translation. This is where you take DNA, copy it into RNA, then you use the RNA after you've edited it, and you read it in sections of 3, which are called codons, and you use that to make proteins.